Hi everybody, let's study meiosis together. So what is meiosis? Meiosis is a process where a single cell divides twice to produce 4 cells containing half the original amount of genetic information. So as you can see, this is the parent cell and it's going to divide twice, right? So this is 2n, 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 and as you can see here, there are the 4 haploid granddaughter cells, right? So during meiosis, one cell divides twice to form 4 daughter cells. These 4 daughter cells only have half the number of chromosomes from the parent cell, and they are haploid. They're half the number, right? So here half, here half, here half, here. So 4. Meiosis produces our sex cells or gametes, egg and sperm cells, right? So mitosis is somatic, like body cells, and meiosis is egg and sperm cells. All right, so what are the steps and stages for meiosis? So it starts with interphase, right? And then we have Prophase I. And here is Metaphase I. And then we have Metaphase I. And then we have Anaphase. And this is Telophase. And now we have a second round of division, which starts with Meiosis II. And again, we have from Prophase. So as you can see here, we have interphase, the cell starts to divide in Prophase I, right? So as you can see, we are already starting to get aligned at the equator, right? And as you can see that we have spindle fibers, we have microtubules, right? So here we already have a plate, right? On the equatorial plate, you'll actually see chromosomes are all lined up, right? In Anaphase I, you will see chromosomes getting pulled, right? And as you can see in telophase, we are starting the pinching process, and from the pinching process, we have cytokinesis. As you can see, we have 2 daughter cells, right? And each have a pair of chromosomes, 2n, 2n. So now in Metaphase II, as you can see, they're actually starting to line up again, right? At the equator, and in Metaphase II, we already have again a lineup, and then anaphase, right? As you can see, the mitotic spindles are pulling the sister chromatids apart, and now the sister chromatids are all lined up, right, at the opposite poles. And now the pinching process starts at telophase. We have 4 haploid daughter cells, right? So 1 chromosome from each, as you can see, 1 here, 1 here, 1 here, from here, right? All right. So what happens in meiosis? Meiosis can be divided into 9 stages. These are divided between first time the cell divides by meiosis I, second time it divides, which is called as meiosis II. The 4 daughter cells produced by meiosis are haploid, meaning they have half the number of chromosomes as a parent cell. When a sperm and egg cell combine, they create a diploid cell called zygote, right? The zygote then undergoes mitosis to develop into a new organism. So now as you can see, from meiosis, we're going towards mitosis, right? So because now egg and sperm, they came together, and now we have somatic cells, the body cells, and we have a new organism, right? So meiosis I is a type of cell division that reduces the number of chromosomes in a cell by half, right? So this is just half. It's the first of the 2 divisions in meiosis, which is a process that occurs during sexual reproduction, right? So as you can see, this is Prophase I, right? Pair of chromosomes, and there are 2 spindle fibers, as you can see, right? And here we have 2 spindles, right? And we have mitotic spindles, and as you can see, microtubules. They're literally pulling chromosomes at the metaphase, and then slowly they'll start pulling them towards the opposite poles. And now we have telophase I, right? And we have cytokinesis, where the pinching happens and they separate into half, right? So from 1 we have 2, as you can see, right? So we have 1 here, 1 here, we have 1 here, 1 here. And as you can also see, they actually went through a process of recombination, right? So they just had a process of genetic crossover. All right, so interphase, the DNA in the cell is copied, resulting 2 identical full sets of chromosomes, right? So here, during the interphase, we have DNA in the cell is copied, resulting 2 identical full sets of chromosome. Outside of the 2, sorry, outside of the nucleus are 2 centrosomes, right? So this is nucleus, and these are the two centrosomes containing a pair of centrioles. As you can see, these drum shapes are centrioles. These structures are critical for the process of cell division. They're very important, right? As you can see, these are esters. Esters are pre-microtubules, right? So during interphase, microtubules extend from these centrosomes. Microtubules are going to extend from the centrosomes, right? Interphase esters are microtubule structures. These are esters. They are microtubule structures that span the cell during interphase. The period before the cell division, they are important for determining the cell's geometry and the site of cell division, where does the cell division occur, and also the cell's geometry, structure, of the cell. As you can see, this is the nucleus, this is a nuclear envelope, as you can see. This happens to be the plasma membrane. These are chromatin dispersed chromosomes or chromatins. All right, so prophase I, the copied chromosomes condense into X-shaped. So as you can see, these are copied chromosome condensing into X-shaped structures that can be easily seen under a microscope. You can see them under a microscope, right? Each chromosome is composed of two sister chromatids containing identical genetic information. So as you can see here, each chromosome is composed of two sister chromatids, two sister chromatids, right, containing identical genetic information. The chromosomes pair up, so both the copies of chromosome 1 are together, both copies of chromosomes 2 are together, and so on. So they're pairing up, they're coming together during prophase I, right? The pairs of chromosomes may then exchange bits of DNA in the process called as recombination or crossing over. So as you can see, the blue was recombinated with the red, right? So as you can see, this little part exchanged, the crossing over, and the red part from here went to the blue, right? So that's called as recombination crossing over. At the end, this is exactly why we can actually see variations, right? All right. At the end of prophase I, the membrane around the nucleus in the cell dissolves. So this nucleus is going to dissolve away, releasing the chromosomes. The chromosomes will be set free after recombination. The meiotic spindle, consisting of microtubules and other proteins, extend across the cell between the centrioles. The meiotic spindles, consisting of microtubules and other proteins, extend across the cell between the centrioles, right? So as you can see, the meiotic spindles are actually extending across the cell between the two. These are extensions. From esters, we have microtubules, and then microtubules play a very important role in metaphase I. This is exactly why in prophase they start extending. The chromosome pairs line up next to each other along the center of the equator, right? So line up. As you can see, this is a lineup. And this is exactly why metaphase, anytime when you see a metaphase, remember the plate. You're lining up your carrots in a row like that. The centrioles are now at the opposite poles. Now these centrioles, 1 and 2, are on the opposite poles with mitotic spindles, right? So as you can see, these are mitotic spindles, and the centrioles are now opposite poles of the cell with mitotic spindles. Very soon they'll start extending and pulling the chromosomes apart. The meiotic spindle fibers attach to one of the chromosome of each pair. So just one here and one here. They will attach to it and then pull them apart. Anaphase I, okay. The pair of chromosomes are then pulled apart. So now they're pulled apart by the meiotic spindle, which pulls one chromosome to one pole of the cell and the other chromosome to the opposite pole. So they're getting pulled in opposite pole. In meiosis I, the sister chromatids stay together. Sister chromatids are going to stay together. This is different to what happens in mitosis and meiosis II. The sister chromatids are going to stay together in anaphase I, okay, in meiosis I. All right, telophase I and cytokinesis is very important. The chromosomes complete their move to the opposite poles of the cell. So as you can see, the chromosomes are pulling apart in the opposite poles. At each pole of the cell, a full set of chromosomes gather. So as you can see, at each pole of the cell, the full set of chromosomes are going to gather. A membrane forms around each set of chromosomes to create two nuclei. So a membrane is going to form, to create two nuclei. The single cell then pinches in the middle to form two separate daughter cells, each containing a full set of chromosomes within a nucleus. So the two daughter cells are going to be formed containing full set of chromosomes within a nucleus. This process is known as cytokinesis. This process is known as cytokinesis, the pulling apart. Cytokinesis is like the end of division, but because meiosis has two, this is actually going to be the start of the second division. Alright, the meiosis II is a type of cell division that produces haploid gametes or sex cells for sexual reproduction. It is the second part of a two-part process called meiosis. So as you can see, this is Prophase II, two spindles, mitotic spindles. We have a pair of chromosomes here, pair of chromosomes here. Alright, so two of them are starting to line, or they are getting lined up in Metaphase II. This happens to be Anaphase II, where two sister chromatids are pulled apart, right? As you can see, this is Telophase II, where you have one from each parent, right? Prophase II, now there are two daughter cells, each with 23 chromosomes, 23 pairs of chromatids, right? In each of the two daughter cells, the chromosomes condense again into visible X shape. So now the condensation happens again. X-shaped structures that can be easily seen under a microscope. You can see them under a microscope. The membrane around the nucleus in each daughter cell dissolves away, releasing the chromosomes. The membrane around the nucleus in each daughter cell dissolves away. The centrioles duplicate. Now, from one centrioles, you have a second one. The meiotic spindle forms again. All right, so these meiotic spindles are going to form again. Metaphase II, in each of the two daughter cells, the chromosomes pair of sister chromatids line up end to end along the equator of the cell. So in Metaphase II, they're going to line up. The centrioles are now at the opposite pole in each of the daughter cells. So centrioles are now at the opposite poles, and they are going to be in opposite poles. Meiotic spindle fibers at each pole of the cell attached to each of the sister chromatids. Okay, so these are chromosomes aligned along the center of the cell. This happens to be in the middle, the center of the cell that is called as a metaphase plate. Spindle fiber attached to the centromere. The one in the middle is the centromere, this little circle. This is exactly why the spindle fibers are going to attach and they're going to pull them apart, okay? And this is exactly why we'll just see this part pulled on one side, this part pulled on another side. Anaphase II, the sister chromatids are then pulled to the opposite poles due to the action of the meiotic spindle. The separated chromatids are now individual chromosomes. Individual chromosome, individual chromosome, individual, all right? Okay, now telophase II and cytokinesis. The chromosomes complete their move to opposite poles of the cell. At each pole of the cell is a full set of chromosomes, right? So a membrane forms around each set of chromosomes to create two cell nuclei. So as you can see, there's going to be a membrane. This blue line is a membrane in telophase II, right? Now slowly they're going to start pinching and they're going to separate. This is the last phase of meiosis. However, cell division does not complete without another round of cytokinesis. Once cytokinesis is complete, there are 4 granddaughter cells, each with half set of chromosomes, which are haploid. In people with XY chromosomes, these are 4 cells, are all sperm cells. So all the 4 cells, in people with XY chromosomes are sperm cells. In people with XX chromosome, one of the cells is an egg cell, while the other 3 are just polar bodies. So one is going to be an egg cell, the functioning egg cell. Small cells that do not develop into eggs. Polar bodies are like small cells, they do not develop into eggs. So out of 4, only 1 is useful for an egg cell. Right. So telophase II, it starts pinching, they separate, and cytokinesis, which is like the end of cell division. You have 4 cells, 23 chromosomes, 1 N, so 1 N, 1 N, 1 N, 1 N, 4, right? Each cell genetically differs from each other and the parent cell. Why? Because we had twice the meiosis, twice the cell division. We also had twice chromatids separating each other. Very important part of variation. We also saw recombination and crossing over taking place, right? Okay. All right, so that's it. Meiosis, mitosis, they're not that complicated if you understand the mechanism and steps of it. All right. Very soon I'll be creating another video that shows what is the difference between mitosis and meiosis, and that's where you'll get more of the information, right? So when you compare to each other, somewhere you will realize that my understanding is becoming more clear. You'll have clarity in the process. All right. My suggestion to everybody who's trying to learn them, try to create diagrams and write around the diagram the steps so you can remember if you want to take notes. Okay. All right, so subscribe, like, share, and comment to my channel. If you have any questions, go ahead and just comment them. I do not respond to emails. I do not provide any private tuition. Everything on my channel is for free, So just comment and ask your questions, and I'll be happy to answer them. Okay, bye.